Good evening, everybody. Thank you so much for giving me chance to meet such a good audience. I could already see the questions which is coming from the audience. So today, at the second part, you're going to see. So far, you have already got the background on where does the bioinformatics started and different kind of data sets and how it has been can be handled. What are the recommendations and all those things. So what we are going to cover now, or probably what we are going to see now is currently the need of our the informatics field is automation, right? So being a biologist, starting to know about the basics, and then slowly you should be down the line able to come up with the automation. That's what the ultimate goal is. It's not only the research field or the one of the good question asked by one of the like say the attender about whether in the hospital or in the research institute or in the academic anyway. Finally, the automation comes in picture once you have more data in place, right? So that's what title also stands for the same thing: A for automation and B for biology. So our theme is basically we are focusing on the informatics. Slightly, my interest is on the clinical side. Why? Because I have knowledge on the clinical background specifically, right? So what are the things we are going to cover here? Different kind of data sets. What are the kind of data set currently you have, even if you are not having any machines in hand, right? And what are the sources and resources, and how does the analytical and logical thinking goes? And what is mean by best practice guidelines? What are the policies or compliances, and what does it means? Why it is very important? And finally, we are going to say that we are going to see how the data flow or the archival methods are, right? So first, let's see the data sets. We all know the data sets when I meant here with respect to clinical, like with respect to clinical research or the clinical labs or the hospitals. Human as a human, we have multiple organs. There are difference in men and women, as you can see here. These are all the important organs, right? Say heart, lung, stomach, liver, kidney, bladder, right? And brain, eye, nose, lips. So these are all the parts where generally we should be very careful with. And most of the people who are either a healthy one or probably who have a disease, they that the main clinical research is going upon these kind of major areas only. And one of the very important area which I can suggest is the cancer and the surveillance and MEMR. The reason for saying all these three things are basically the data size, or probably these are all the major factor where even the public services are very much interested in, right? So these are all the data which is basically used for different groups, teams, or even in the hospitals or in the labs for the diagnostic purpose. So, okay, let me get rid of this floating. Yes, hope it is very clear. So when I say data sets, right, it can be different. Some people wants to analyze the data in the single cell level. Some people want to do the analysis on the single base, in the sense ATGC level. And recently there was a question on how do we do the analysis on theoretical science, right, where the biophysics comes in picture, atom level they want to go for. And there are people who want to work on protein or DNA, which is structural biology. And people wants to go for a neural network and pathways, which is basically gene level. So when I say the data sets or the type of the data, it's based on which question you are asking, what do you actually want to study, right? So it's overall when we say it's a biological science, but in detail when you dig in more or on the interest as well as the scope of the study, it little bit varies on which groups it belongs to. This is also was been asked before. What has been the computation and bioinformatics difference? So it's not only these terms are interrelated. Rather, I would say even biostats. So those people who use the biological data sets and they apply statistics on it to come to the to interpret some data sets and then come to the conclusion. Biomedical engineering, where obviously they will be knowing they have a mechanical background so that they can repair all the medical equipments very well, right? They are basically mechanical engineer who knows how the modules are placed in the probably the medical item or biological items. When I say items, it means machineries. And finally comes bio IT. These are all the field which is interconnected, I would say, right? Bio IT, when I meant, it means the IT person who is handling a biological data, who is very much aware of how much the RAM is, how much the storage is needed, how much HPC system is needed or not, how much clusters has to be distributed. So all those IT related background, as well as currently, if you see, most of the products or probably the softwares are getting developed in cloud environment. So in that case, bio IT is having a high scope and probably I would say current demand, right? So let's see the sources as well as the resources. So when I say sources as well as resources, it means it is current. It's not like we are in olden, like say olden days where we don't know where to get the data, where things are placed in. So currently, if you see NCBI or NIH basically has all the modules. You can see the list over here, which based on the questions you want to answer, you can select the respective tabs and you can get the data downloaded. Where are the data are coming from? Basically, it's been submitted with the publications by the researchers mostly. So as you can see, this is for submission with the manuscript, and this is for the users who want to utilize the data set, right? So before even going to touch the data set or probably use a data set, there has to be a small checkbox which has to be checked in every user's. Is it public repository? Yes. Is this protocol is established? And what is the size of the data set? Is it good enough? It's not like having only 5 samples and then coming to the conclusion, because statistically it is really not good enough to support the evidence. So in that case, the data size should be good. And which group is been studying, like they study groups. If it is a random one, the odd one should be removed totally. In that way, these are all the small checklists which has to be checked, and quality of the data, obviously. So why I'm saying, because when you do validation using the data which is open source or available, right? Based on that, you get your results of test modules, right? So in that case, these are all check box which has to be in place before even finalizing on any kind of data set to be chosen. And currently, we can't blame on anyone saying that I'm not getting the data. We are in 2023, and all the data are in digital format, and it is available and it's freely available. That is the main thing which I would like to highlight, right? And resources, the previous speaker have already very nicely portrayed in a different format. I'm reiterating the same thing. It's one is computational need. Another one is, when I say computational need, it means how much RAM, how much storage, and like say which kind of the computation. When I say server is good enough to do this task, we should be knowing forehand. And probably the best option is, if any publication which speaks about analysis or probably the product development or the algorithm development or the validation studies, you feel free to write to them and ask first. I would like to do same kind of study, but my study is a little bit different from yours, slightly different. But I would like to know what is the computation facility you had. So computation facility comes in picture first, and then comes the software as well as the platform needed. So what is meant by software and platform needed? Currently, if you want to do an analysis, there will be a middle point, A, B, C, D, A to B, B to C, C to D, right? So in that case, we should be knowing what are the softwares are needed to go from A to B. A is your input, B is your output, and then this B will be utilized as an input in the next module. In that case, we should be knowing what are the softwares are needed and what are the platforms it supports. For example, if I say for NGS, if you have a Linux system, it is good enough. If you are using Windows, it is kind of not a good platform to suggest, right? So in the same case, if you are handling a protein, there is no meaning of going for Linux, because most of things can be done using Windows system itself, right? Protein level works. So in that case, we need to know what do I have, like say what kind of data set I have, which softwares are good and which platforms are good. Implementation of the skill set. So this comes by practice. There is no shortcut for it. Skill set should be developed day by day by understanding the problem and brainstorming. So basically the skill sets can be learned. Currently, the previous days, it is just only textbook. You will be referring to textbook and then you have to try it your own. Currently, you have many online resources. There are many YouTube channels which teaches many different things, including the analysis, right? So in that case, it's easy to learn. I think one of the person was asking about Galaxy also. So in that case, it's an open source. The only thing which you need to be careful is putting a clinical data in the Galaxy is kind of, Galaxy is a cloud computing bio NGS data and there's a software. Putting the clinical data there is kind of risky. Otherwise, it's good enough to use that, right? So we should be knowing the places where you can do the like say analysis in online itself. It's not like every problem has to be cracked in by using the facility, establishing a facility. Rather, there are cases where if you know the online softwares which is available for solving specific bioinformatics problem, that can also be utilized. Estimated time frame. So if before even accepting any projects or before even planning any study, currently someone has asked about the project, right? So we should be knowing clearly the time frame. If I'm going to do this task, having all this software based on the skill sets which I have, how long it is going to take. So the time frame which has to be like say probably estimated in a better way. And finally, the manpower to complete any individual task, how many people are needed and how much time it's going to take. So these things which has to be followed strictly. Followed in a sense, knowing, you should be knowing, or at least we should be taking help from others to know the things.